What does the plasma membrane do? What does the plasma membrane do? It mediates the entry and exit of substances. What does the cytosol What does the cytosol do? That's the liquid. It's composed of water, solutes, suspended particles, lipid droplets. And it's the medium in which many of cell metabolic reactions occur. The centrosome. What does the centrosome do? Used for growth of the mitotic spindle and microtubule formation. Growth of the mitotic spindle and microtubule formation. What does the ribosome do? Those little dots. On rough ER. Protein synthesis. What does the rough ER do? It synthesizes glycoproteins. The rough ER synthesizes glycoproteins. What does the smooth ER do? What does the smooth ER do? It looks like coral and it synthesizes fatty acids and steroids. Fatty acids and steroids. It stores and releases calcium in muscle cells. What are the two things it do? Synthesizes fatty acids and steroids. Secondly, stores and releases calcium ions in muscle cells. What does the Golgi complex do? Sorts and packages. Sorts and packages. Proteins. What does the lysosome do? What does the lysosome do? It digests the contents of endosomes and transports the final products of digestion into cytosol. It also digests worn out organelles, which is called autophagy, and digests entire cells, which is called autolysis. The nucleus contains genes and controls cellular structure. What does the nucleus do? Contains genes. What does a cytoskeleton contain? Microfilaments, intermediate filaments, and microtubules. Notice the centrosome in the middle. What three types of protein filaments are in the cytoplasm that are called cytoskeletons? Microfilaments, one starting with I and the one starting with M. What forms of active transport do we have? We have the sodium-potassium pump that expels sodium ions and brings potassium ions into the cell. It expels sodium ions and brings potassium ions into the cell. Firstly, the first process of the sodium-potassium pump is that 3 sodium in the cytosol bind to the pump protein. This triggers the hydrolysis of ATP into ADP. This changes the shape of the pump protein. Two potassiums are bound to the protein. This binding of potassium triggers the release of it. The different concentrations of sodium and potassium are crucial for maintaining normal cell volume and also for the ability of some cells to generate electrical signals such as action potentials. Can you turn it back on, please? I need that on. This is charging. Thank you. Leave it. Secondary active transport. The energy stored in a sodium concentration gradient is used to drive another substance across the membrane against what? Concentration gradient. If two substances cross the membrane at the same time, like in secondary active transport, they are either symporters or antiporters. Antiporters move the substances in what? Opposite directions. Symporters move the substances in what? Same direction. This is facilitated diffusion, and that's what glucose uses. Facilitated diffusion. Glucose binds to a glucose transporter And as the glucose transporter changes in shape, glucose passes through the membrane and is released in the cytosol. Endocytosis. Endocytosis. When all receptors are full, it engulfs them to look like this, which is called vesicle formation. It then fuses with the endosome. The uncoated vesicles then fuse with the endosome. The receptors are recycled to the plasma membrane. The transport vesicle sends it to the lysosome for degradation, and the lysosome uses its digestive enzymes to digest it. This is called the clathrin coated pit. In exocytosis, exocytosis can either be constitutive or regulated. If it is constituted, the molecule secreted needs to be in constant levels. If it's regulated, it's in response to a stimulus, secreted only when needed. An example of regulated is the secretion, two examples, the secretion of neurotransmitters and the secretion of glucose transporter. A constitutive secretory pathway is here. Newly synthesized plasma membrane lipids, newly synthesized plasma membrane proteins, and the regulated secretory pathway is that the trans-Golgi network secretes vesicles storing the needed secretory proteins. Back to secondary active transport. The symport, as we said below, is a process in kidney. And antiporter, as sodium comes in, potassium goes out. And a uniport is a simple glucose transporter. You can see some of those here.
You can use them.
Go, please. You've interrupted me twice, Daniel. Go. Jaden, go. Okay, turn mine off. It's fine. And go. Plug it into another room. Antiporters carry substances across the membrane in opposite directions. Symporters carry two substances across the membrane in the same direction. Mitosis. What happens in prophase? This is prophase. What happens in metaphase? What happens in anaphase? What happens in the last phase where the cleavage furrow completely separates into two nuclei? What's the name of this starting with C? What's the name of this? What's the name of this? It's integral and peripheral. Is ligand gated channels integral only? What do linker proteins do? What is this called? It catalyzes reaction. And is it just integral or is it integral and peripheral? It transports a specific what? Substance across the membrane. What's the word that describes polar heads and non-polar tails? What's the word for that? What's an example of a concentration gradient? Something are more concentrated on the outside of the cell. Back to interphase. What is G1? Cell metabolic rate increases. What is S phase? DNA replication. What is G2 phase? conclusion of metabolic increase. This is the entry into the mitotic division. First of all, we have early prophase. What condenses and shortens? Something starting with C condenses, and then they shorten into visible. In late prophase, the nucleolus and nuclear envelope disappear. And we know that each centrosome moves towards opposite ends of the cell via mitotic spindle. After prophase, we say PMAT. PMAT. So we know it's metaphase. How would you describe the metaphase? What lines up? The centromeres line up at the center of the what? Mitotic spindle. And what's the region called? Metaphase plate. PMAT. What's the next phase? Anaphase, early. First thing is that the splitting and separation of centromeres. The late one is movement of the V-shaped chromatids towards opposite poles of the cell, and that is late. What's the last one? PMAT. That's right, telophase. The identical sets of chromosomes at opposite poles of the cell uncoil and revert to their thread-like chromatin form. New nucleoli appear. The last thing that occurs is cytokinesis, which is the division of a parent cell's cytoplasm and organelles. How is mitosis and meiosis different? In meiosis, it's just the ovaries and testes that undergo the process, and it's purely for reproduction. Cell undergoing meiosis is diploid. The chromosome number is halved in the products of meiosis. In mitosis, the chromosome number per nucleus is maintained. So the cell undergoing mitosis can be diploid or haploid. The daughter cell ends up being genetically identical with the parental genotype, whereas in meiosis, there's variation. Lastly, adherens junctions. It contains transmembrane glycoproteins. It's common between cells that line the GI tract. It contains plaque. Ones that are similar to adherens is desmosomes. But it also connects to the intermediate filaments. Desmosomes are in the skin and cardiac muscles. Half a desmosome is a hemidesmosome, and it connects cells to what? To the basement membrane. It can be found in where? Some cell junctions form fluid-tight seals. What are the ones that form fluid-tight seals? Tight junctions. Tight junctions have strands of transmembrane proteins. And these are also called transmembrane proteins. The plasma membrane is fused with a strip of proteins. Where can it be found? Somewhere where there's fluid-tight seals. Cells that line the GI and bladder. Which cells anchor cells together? That's right, they're hemidesmosomes and desmosomes. Which cells act as channels which allow ions and molecules to pass from cell to cell? That's right, gap junctions. Gap junctions. Gap junctions.